Hey guys, welcome back. If you remember last time, we were talking about DNA electrophoresis and how we can use it to separate nucleic acids according to their molecular weight. I want to continue that conversation and talk about how we can use electrophoresis to separate proteins as well. So with the same idea as DNA, we have protein electrophoresis, all right, where we can separate, we can put a collection of proteins into electric field, and those proteins will migrate or move through that electric field depending on their particular charge, their size, and even the shape of the protein. So before we get started or too far ahead of ourselves, let's step back a little bit and remind ourselves of how proteins kind of work. So from the building blocks of amino acids, we get these primary structures, which is just a sequence of these monomers of amino acids. And then as these amino acids start to fold and interact with one another, these polypeptide chains then start to create these secondary structures called alpha helices or beta sheets, as shown here. So then these beta sheets start to fold and 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 collapse on one another and fold into these tertiary structures. And this is starting to give it more of that three-dimensional shape. All right, and then finally we end up with a complex protein and a quaternary protein structure, and these can be different structures depending on their need throughout the body. All right, let's focus on the primary structure here for a second. The primary structure is made up of these polypeptide chains, all right? And this chain consists of a bunch of amino acids that are bound to one another. And each amino acid, all right, contains this amino group, all right, and on a carboxyl group. Each amino acid also contains a side chain, and this side chain is what determines the properties of each of our amino acids that make up these polypeptides. There are 20 amino acids, and each of the amino acids are classified by their particular R group. So we can have acidic amino acids, we can have basic amino acids, we can have non-polar amino acids, and we can have polar amino acids. And so here's a chart that I put together for you guys that lists the non-polar, all right, amino acids, and then the polar and neutral amino acids, the polar acidic and the polar basic amino acids. So the number of amino acids and which amino acids, whether polar or non-polar, those will determine the structure of the protein along with its overall net charge in the primary structure. However, proteins are not naturally found in their primary structures. Proteins are globular, especially in the secondary and tertiary structure. This is due to those disulfide bonds that you can see here, and the hydrophobic and hydrophilic properties or interactions with its aqueous environment. So in order to get a protein to its primary structure, into its monomer form, we must do something to break, all right, those disulfide bonds, all right? And so we introduce a reducing agent, all right, that's going to help or denature, all right, these proteins or break these disulfide bonds. To use electrophoresis to determine a protein's molecular weight, all right, it's necessary to denature the protein and to introduce a charge onto each of the actual peptides. And we do this with two chemical agents. One is sodium didotocell sulfate, all right, and this is a detergent, all right, that possesses hydrophobic ends and hydrophilic ends. All right. And then BME, which is a reducing agent, which will actually break those disulfide bonds and reduce the protein down to its primary structure. So if we introduce a little SDS and some BME plus some heat, we can get a linear denaturing of the actual protein. A technique that is often used to determine the molecular weight of different proteins is a technique called SDS page. All right. So here we'll start off with the globular form of the proteins and denature the proteins using that reducing agent BME. All right. And then we add the SDS, which will apply an even charge across each of the proteins. And then depending on the charge to mass ratio, all right, we can then put our proteins into a gel, run that through the medium, all right, and the proteins will be separated by their size. The next slide will show a video that will go into a little bit more detail about the SDS page.
The first step in many protein analytical methods, including Western blot analysis, is to separate the proteins in a sample by gel electrophoresis. The most widely used protein electrophoretic technique is sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS page, in which proteins are denatured and evenly charged by the ionic detergent SDS, so that the proteins migrate through the gel based only on size. Polyacrylamide gels can be poured in the lab by combining various concentrations of acrylamide and bisacrylamide with the cross-linker ammonium persulfate and TIMID, which catalyzes the polymerization reaction. Precast polyacrylamide gels are also available, with various acrylamide concentrations and gradients. Let's look at performing SDS page using HeLa cell lysates and precast gels. After adjusting protein levels so that all lysates have a uniform protein concentration, combine the lysates with a sample buffer that includes SDS, glycerol, a tracking dye such as bromophenol blue, and a reducing agent such as dithiethrietol, beta-mercaptoethanol, or TSEP. Heat the samples for 3 to 5 minutes to denature and reduce the proteins, and then cool them to room temperature. Place a gel into an electrophoresis unit, and fill the upper and lower buffer tanks of the unit with a running buffer made of tris, glycine, and SDS to enable proper flow of the electric current through the gel. Then load the protein samples into the wells in the gel. Because the glycerol in the loading buffer is heavier than the running buffer, the sample should easily drop to the bottom of their respective wells. The volume loaded into each well can range from 1 microliter to over 40 microliters, depending on the protein concentration, well size, and gel thickness. Also, load a separate well with a molecular weight marker to assess the relative molecular weight of the target protein. Once the samples and marker are loaded, connect the electrophoresis unit to a power source, making sure that the cables are correctly connected to ensure correct protein migration. Then apply an electric field at the appropriate voltage or current, which depends on the type and number of gels being used. Bubbles appearing around the cathode indicate that the current is flowing. Once the die front reaches the bottom of the gel, turn the power off and unplug the cables from the power source. Remove the gel cassette from the electrophoresis unit and carefully separate the gel from the plates. The gel is now ready for Western blot, Kumasi stain, or another protein analytical method.
One of the big advantages of using SDS page is that because we've reduced those proteins down to their monomer and to their primary structure, this is going to nullify any effects that a complex, more globular form of a protein might have on the mobility. And this is going to allow us to separate based on that charge to mass ratio. Right? So the larger subunits are going to have are going to be slower, all right, and the smaller ones are going to move quicker through the medium. Right? This also allows us to separate subunits of proteins that are much more complex. So if you have an alpha unit, beta unit, it will separate those units from the proteins as well. Native page assay allows us to look at the proteins while they're still in their globular form. All right. And so for native proteins, the mobility depends on the net charge and that frictional coefficient, or their size and shape, basically. And the net charge depends on the amino acid composition and the charges of any ligands that might be actually bound to the protein. The net charge is always a function of the pH, and so therefore if we choose a pH carefully, different proteins can be separated. All proteins will be positively charged at a low enough pH and will be negatively charged at a high enough pH. So this means that at some particular pH, each protein will be neutral, right? And this pH is what we call the isoelectric point, right? And this is the pH at which the protein has zero mobility, right? And native pages are useful for being able to get information on protein charges, obviously, and separate proteins that have the same identical molecular weight but may have a different charge. And it also allows the proteins to keep their activity, such as ligand interactions, protein-to-protein interactions, things like that. And it helps us determine different protein conformations, isoforms, coniforms. So here's just a side-by-side comparison of SDS page and native page. Remember, the native page separation is going to be based on the charge, the size, and the shape of the proteins, whereas in the SDS page, we're only going to separate based on the actual molecular weight. Isoelectric focusing is a technique that allows us to use the property that we just talked about in proteins of that isoelectric point. Remember, this is the point at which a protein has no net charge and will not move within that electric field. And the way that this works is that we take buffers of different pHs and we use that to establish a pH gradient within the gel. And so at high pHs, typically we'll put the high pH at the negative electrode and the low pH at the positive electrode. We can then introduce our samples into the high pH side near the negative electrode, and then the negative charge proteins will move toward the positive electrode until one of those proteins reaches its isoelectric point, all right, and then it will stop. All right, it'll stop moving. So similarly, if we were to start the proteins at, say, the low pH side near the positive electrode, each of the proteins will then move to its isoelectric point and stop as well. This is a technique that's often combined, all right, with the SDS electrophoresis, and creates a very, very powerful separating tool that we'll talk about a little later on. So serum is the liquid part of our blood that's found once we separate it from the red blood cells, white blood cells, and platelets. A serum protein electrophoresis, or an SPEP, is a clinical laboratory technique that's used to determine the levels of some types of proteins in our blood sample. And a doctor might order this to help diagnose or monitor a variety of different diseases or disorders that will affect these particular proteins. One of them is the albumin protein. Albumin is one of the most abundant proteins in our body, and it transports substances throughout our body and plays a role in tissue growth and repair. Another protein is a alpha-1 antitrypsin globulin protein, all right? And this is produced by the lungs and the liver and can increase with inflammatory diseases. An alpha-2 globulin all right, is a class of protein which has many functions in the body and is involved in inflammation. A beta globulin is a protein used to move substances, support immunity, and increases in number if you are diagnosed with something like multiple myeloma and conditions like high cholesterol or atherosclerosis. And then finally, we have these antibodies or gamma globulins, and these support our immune system, right, usually typically found in immunity, and can increase with multiple myeloma, as well as some of those autoimmune conditions like rheumatoid arthritis, things like that. These can also show a broad increase just dealing with particular infections. So in the previous slide, I showed you guys what a typical healthy SPEP might look like. In this slide, I'm just giving you guys some idea of what you can expect in, say, an abnormal SPEP. All right, so sometimes increased albumins may indicate dehydration. However, if we have decreased albumin, we can have any one of these particular conditions seen under this. Same thing if we see like an increase in our gamma globulins, okay, several different types of diseases, like I said, rheumatoid arthritis, or any of these type of chronic infections can also be contributed to seeing an increase in gamma globulins. Some special conditions I would like you guys to be familiar with when looking at different SPEPs. One of them, a bisalbuminemia, is sometimes a condition that can be inherited, but is typically characterized by having two peaks in the albumin region. So you can see here in this particular graph, you see two large peaks in the albumin protein region, indicated here by the P, and seeing the two peaks there. The nephrotic syndrome is a collection of symptoms that can be seen due to kidney failure or kidney damage. All right, and these are characterized by seeing increased peaks in the alpha 2 region and in the beta globulin region. The gamma globulin region is given special attention or special focus because it has everything to do with our immune system. And so abnormalities in this region are characterized by two different things. All right, you can either have a monoclonal increase in that gamma region, as seen in this figure, where we have just a single increase, all right, or a strong monoclonal increase, which we see here in this particular figure. And those can indicate certain things like multiple myeloma, or which we know is a bone marrow tumor, or this other type of disease. A polyclonal increase would indicate multiple regions within our gamma globulin region, such as in this figure where we see kind of three peaks here, or one large broad peak. So typically if we got one large broad increase, that might be a systematic or an acute inflammatory response by our immune system. The immune system is made up of several types of cells that work together to fight infections and other diseases. Lymphocytes are part of that immune system, and they're made up of T cells and B cells. So when a B cell responds to an infection, it ends up maturing and changing into what is called a plasma cell. All right, and this plasma cell goes on to make antibodies called these immunoglobulins. and that helps our bodies attack and kill germs. Um, these plasma cells are mainly found in bone marrow, and in an SPEP, monoclonal increases in that gamma region can help us diagnose different plasma cell disorders such as multiple myeloma, monoclonal gammopathy, and light chain amyloidosis. Multiple myeloma is a condition in which these plasma cells become cancerous and grow out of control. And because cancer cells don't mature and then die like normal cells do, they end up accumulating and eventually overwhelming the production of these healthy cells. And in bone marrow, myeloma cells end up crowding out healthy white cells and the red blood cells, which ends up leading to fatigue and the inability to fight infections. The myeloma cells continue to try to produce antibodies, just like the healthy plasma cells do, but the myeloma cells produce abnormal antibodies that our bodies just can't use. And so instead of the abnormal antibodies or these M proteins, they end up building up in the body and causing problems such as like damage to our kidneys and stuff. Cancer cells can also cause damage to the bones, which can increase the risk of different broken bones as well. Monoclonal gammopathy can kind of be considered the precursor to multiple myeloma, and which these cancerous plasma cells end up producing many copies of the same antibody, and so the antibody region or that gamma region ends up with a large M spike. And so although it should be noted that people with multiple myeloma have a monoclonal gammopathy, not everyone with monoclonal gammopathy has that multiple myeloma. Because of the great degree of difficulty in distinguishing diseases in just a SPEP test, doctors will often order a second test on the urine, and what we call a UPEP test, or urine electrophoresis. And they look very similar to a serum electrophoresis. Again, we have all the same regions, except in a urine electrophoresis, we're typically looking for this Bence-Jones protein, which is a spike in the gamma region here. And that spike in the gamma region is caused by the body trying to get rid of these excess antibodies that the body is producing. And so remember, in those cancerous plasma cells, they're producing a lot of unhealthy antibodies, and we're having a lot more antibodies that we need to try to get rid of. And one of the big problems with the antibodies that are produced by those cancerous plasma cells is that they have these too many light chains. And so this is an example of a healthy antibody, which has 2 light chains and 2 heavy chains. However, a cancerous antibody or antibody that's been produced by a cancerous plasma cell will have many of these light chains and not as many heavy chains. And so there's a lot of these light chains floating around, and they end up getting, these proteins end up getting peed out by our bodies, and so we'll end up seeing a spike in that antibody gamma region. If there's enough of these light chain antibodies floating around, the patient might get diagnosed with light chain amyloidosis. Right? And so this is when abnormal body cells are producing, again, too many of these light chains. And these light chains end up getting circulated through our body. Some of them get peed out through our urine and everything, but some of them make it back to our tissues. All right, and the buildup of amyloid in certain organs can lead to a lot of problems for our patients. So, like if we end up with a buildup in the heart, irregular heartbeats, heart can enlarge, these tissues can get larger, inflammation, all right, is prevalent. And then if amyloid is in the kidneys, it can lead to kidney failure or damage at the very least. And if we end up with amyloid in the pancreas, that can be sometimes associated with diabetes. So if you recall earlier, I talked about how we could combine two separation techniques to make a powerful analytical method. So in this slide, I'm just talking about how what if we wanted, or what if we had several proteins that have the same molecular weight? All right, well, if we were doing just a native or an isoelectric focusing run, those with the same molecular weight would show up in the same band. So how can we separate those molecules or those proteins even further? And so the way that we do that is using a two-dimensional gel. And in the next slide, I'm going to talk to you about the actual process of that, and then I'll catch back up with you and discuss it quickly.
Two-dimensional gel electrophoresis is a procedure for separating a mixture of proteins into spots on a gel. In the first dimension, proteins are separated in a tube gel according to their isoelectric points. When a protein in the tube gel reaches the pH where its net charge is 0, it stops migrating and forms a band at that location. Since two or more proteins may have identical isoelectric points, the bands in the tube gel may not contain a single type of protein. The isoelectric focusing tube gel is then laid on the top of a slab gel, which is an SDS gel that separates proteins according to their masses. The proteins within the tube gel are electrophoresed into the slab gel, and then move through the slab gel. Smaller proteins move more quickly than larger ones. In most cases, each type of protein forms a discrete spot that can be identified by staining methods. Individual spots in the gel can then be quantitated, extracted, and analyzed.
So in that two-dimensional gel, we first start off with the first dimension, which is typically an isoelectric focusing technique, which allows us to separate the proteins according to their net charge and that frictional coefficient, or the size and the shape of the proteins as well. All right, and then once we've done that, we've separated them that way. We then lay that that isoelectric focusing strip onto the top, all right, and then run a SDS page, all right, where we use the SDS to then denature the proteins and separate and put a uniform charge on all of our individual components, and then those can then pass through the second dimension, all right, of the gel, and we can then separate those by molecular weight. This is just another slide giving you guys another overview of how to use IEF and SDS page in a two-dimensional gel. And we can see here that usually in 2D gels, they can sometimes look a little confusing, a little complex. We get a lot of information. There's a lot of data points here. So typically we'll use some type of computer, some type of imager to be able to analyze this stuff and analyze the particular spots and the differences between the two techniques. And so this is typically how we read these gels. Here's some of the strength and weaknesses. Strength, we get high resolution. We can really get good separation of different components of proteins, such as isoforms and the variants of it. Good for complex samples as well. And some of the limitations, though, however, it takes a lot of time. You're really only going to see the most abundant. So if you're looking for a very specific protein, it may be difficult. And membrane-bound proteins will not separate well. These are some of the common problems that you guys might run into if you're ever asked to do any kind of gel electrophoresis, you guys can take some time, look at this. I'm not going to go over it in too much detail, but just to give you guys some ideas. And then also I wanted to give you guys just some information on what you might see if you messed up a gel. All right, so here we see a couple of gels that have gone bad, and some of the issues underlying issues for why those gels went bad. That should take us to the end of this. If you have any questions, get in touch with me through email. And again, I'll be holding those office hours during our regularly scheduled time. Thanks. Talk to you later.